The production of this program is made possible thanks to the support of the following and viewers like you. To boldly go where no one has gone before. The phrase was inspired by a 1958 White House pamphlet, Introduction to Outer Space, which highlighted humanity's inherent urge to explore the unknown. It also was the opening to the monologue for Star Trek, where Captain James T. Kirk goes on to say, "These are the voyages of the Starship Enterprise. Its 5-year mission, to explore strange new worlds, to seek out new life and new civilizations." As an aspiration, it drives the space programs of many countries and a growing number of companies. Here's the challenge, however, says Dr. Christopher Mason. Well, we can build machines that can survive in space. The human body was not made for space, at least not yet. Dr. Mason is the principal investigator of the NASA twins study, where 10 separate teams studied an astronaut and his identical twin. Dr. Mason says we observed the physiological, molecular, and cognitive changes that happened to Scott Kelly, who spent a year in space. We monitored the changes in his body compared to his identical twin, Mark, who stayed on Earth. From that study, we're working to mitigate stresses, changes, and new diseases the body may encounter during long-term space flight. This work is the foundation of a 500-year plan to re-engineer biology so that life can be possible for humans on other planets. I invited Dr. Christopher Mason, the Genome BC 2025 Don Ricks Distinguished Keynote Speaker, to join me for a conversation that matters about the human body and its remarkable ability to adapt to long-term voyages to the final frontier. Dr. Mason, welcome.
Thanks for having me. Pleasure to be here.
Where did the genesis of this idea of taking a look at the genome of astronauts and say, okay, we know that just by being there's an epigenetic response, but we need to help you if we want to be able to even get to Mars and stay there for a couple of years.
Yeah, a lot of the concern came from just the observation of the stress on the body, the reports from the astronauts themselves, which we'd heard for decades. We knew that even really since the 60s, that it was very stressful on the body. But we know it was clearly possible for the humans to go into space, to survive in space. And then, as we saw in the twin study with NASA, that even a year in space was possible. And the Russians had even gone up longer before that mission. So we knew even a year and a half was possible. But it did lead to a lot of changes, obviously muscle and bone loss. There was the immune system changes, where a lot of times viruses can reactivate, like oral herpes. You think of cold sores in your mouth. That's very common for astronauts. So clearly there's some changes in the body that some are adaptive, where the body is just accommodating to the fact that it doesn't have to use as much force to push off a wall. But some of them are possibly maladaptive, where the immune system is perturbed in a way that maybe is short-term concerning and potentially long-term a risk, though. So we wanted to understand all those factors. But along the way, we thought, well, this is just mostly observational. What if we could protect them like we do with drugs, or physically protect them? Could we look at the molecular and genetic changes and use it as a guide to help them survive in even longer missions?
So what's neat about that is that you had one astronaut identical twin taking samples and sending you those samples. How did you get them? And then were you monitoring them sort of real time against samples from his identical twin brother here on Earth?
Yes, which was then Mark Kelly, who's now Senator Kelly in the United States Senate. And we were getting samples, basically blood collected in space, dropped into the Soyuz capsule, and then quickly returned to Earth. And within 36 hours, it would land in Kazakhstan, get picked up, repatriated, then brought back to Houston. And we'd get blood samples and hold it in our hand and say, just hours before this was floating in space. So it was really a rapid collection. It's all really thanks to NASA, who had an extraordinary infrastructure to get samples from space and anywhere on Earth really quickly. But then we were still tracking down Mark Kelly on Earth. And there was one point in the study we had more samples from the twin in space than the twin on Earth, just because he was traveling a lot. So
and he was busy being a senator.
Yeah, that's right. He was busy. Wasn't a senator yet, but he was busy being a famous guy, I guess I'd say.
Well, he's an astronaut as well. Yes. That's quite remarkable.
So they're both astronauts. And in a perfect study, we'd have one person who'd never been to space and one person who went to space. But Scott Kelly had been in space about 10 times longer than his identical twin. So it's a pretty big difference. Yeah.
So what did you start to observe well, Scott's in space and Mark's on the ground. What did you very quickly start to say, my, oh my, the human body is adapting to this new environment remarkably quickly?
We saw a lot of things change very quickly, and one of them was, for example, telomeres, which are at the end of chromosomes, got longer in space. Now, normally this is what keeps DNA packaged and actually gets shorter as you age. So we expected it to get shorter because of the stress of the mission, but they actually got longer, and this is working with Susan Bailey. We saw they got longer and then confirmed this across our lab and her lab, and it's a really extraordinary, surprising result. But we also saw a lot of the gene activity. So, of course, we have genes in our DNA in our body, and there's some of them get turned on or turned off for different stressful events or changes in your body. And we could see thousands of them really activated early in flight. So really the body quickly adapted, and with we called fireworks in space. So there's many changes quickly activating as soon as he got into space. And a lot of them were the immune system.
So, okay, but going back to the telomeres, because of course I'm going, well, longer telomeres, hopefully there's less mitochondrial breakdown and when the gene reproduces, therefore I'll stay younger. Was that the case, or did the telomeres just extend, but was there also better sort of reproductive qualities in the replication of the gene?
It was a good question. We saw it was temporary. So sadly, it didn't stay young forever. When we got back to Earth, it went away. And the mitochondria you mentioned also is another indicator of how cells are functioning in their health. And those actually underwent a lot of stress. So we didn't even see a lot of good news for the mitochondria. They also seemed like they were really stressed. But so the telomeres, on average, they got longer, but we also saw a lot of cells with shorter telomeres that looked like they were damaged. So we think it was a bit of the radiation, low dose radiation, that seemed to really be driving this phenotype.
The production of this program is made possible thanks to the support of the following and viewers like you. What do we do? You talk about radiation, and once you move beyond the Earth's protective magnetosphere, like you're now out into space and you're being bombarded by radiation. What is happening there, and what have you learned that maybe can help future astronauts survive what is a more hostile environment?
We think that's the big concern. When in the space station, or even within a few hundred kilometers of Earth, out past the atmosphere, you're still fairly protected. So once we get past that magnetosphere, these Van Allen belts, which really are quite literally a force field around our planet.
So the space station isn't outside of the...
it's still within the relatively protected area. Now, it does get more radiation. It's equivalent to getting a few chest x-rays level every day. It is more than you would get on Earth for sure.
Right.
But relative to what you get in interplanetary space, or even between the Earth and the Moon, once you get that far, you will get a fair amount more radiation, 10, maybe 20 times more radiation that you can start to expect. So it depends how long you're out there. It depends on the solar cycle. Depends on once you're on the Moon, for example, you then suddenly have a moon that's protecting half of your field of view. So you do have protection. Once you get to a planet or a moon, it helps a lot. But in the vacuum of space between planets and moons, it's the highest risk environment.
You're getting bombarded.
Galactic cosmic rays, radiation, and stressful on the body for sure.
Yeah, you get more cosmic rays than you get when you're sitting here on Earth.
Yeah, that's right, yeah.
So you have to start to take all of this into consideration. And you believe that based on the work that you're doing right now, that you can move towards helping the human genome adapt to being able to survive in that kind of environment.
It's possible, and we're working on this in the lab right now. Exactly. So the intriguing thing is we know that humans have always evolved. Like, we are not the same today as we were really millions of years ago. Even the ability to have lactose as an adult is unusual and is a relatively recent change in how our bodies can process food. We think of how humans look different. That's only been over several hundreds of thousands of years, where people migrated around the planet in different hair color, eye color, skin color, different features of our bodies. We're all human, but of course that was enough time for us to all become quite different, and just visibly. So,
and those are primarily epigenetic changes. It's not interference or intervention.
It was just direct. I mean, both genetics and epigenetics. We've seen both are factors. But we think we could, now that we understand genetics much more than we did 100 years ago, even 20 years ago, we can see what's changing. We have a genetic map of which genes are responsible for responding to radiation or the immune system, or what helps you process food or vitamins, for example. So we now have the genetic map in front of us and can say, okay, this is what works well for us to survive in this stressful environment, or why is it that we can have one human being get a disease and one is perfectly healthy? We now can start to map why that is, and once we have that, then that becomes the roadmap of, well, if we're going to a planet with more radiation, should we look at ways we could help the genetic makeup survive in this place?
So how are we going to go about changing the human genome to make sure that it will survive? We already do have that one example of CRISPR babies in China, which the Chinese government even shut down. You're not suggesting that we go to that stage, are you?
No,
of re-engineering the human genome.
You wouldn't likely need to engineer, an embryo stage human unless you had no other option. Like for the only way, so what He Jiankui did in China, which is to make a deletion so the babies could never get HIV, was the goal. But that was with a very early version of CRISPR, which was much less safe, and there was no medical need. The critical part of the ethics of genome engineering for clinical care or for survival on another planet is you have to have some medical need. If you don't have a medical need, then you're just doing research and just playing around. But the medical need possible would be if we send people to Mars, for example, and we've been sending them there for decades, and it looks like there's just too much radiation. And even if they go underground and there's still too much radiation, too much stress, that's the point where you could start to wonder and even deploy an embryo that would be modified to survive on Mars. It just maybe could be modified. That's the genetic modification. The other option is you can do epigenetic modification. Maybe we don't need to add genes or take away genes. You could just turn up higher the genes that help you repair DNA. And that's another work we have in the lab where we can just toggle, just tweak like kind of the knob, just turn things up a little bit, which is probably safer and also more reversible. So I think, but we're looking at both options in the lab right now because we don't know how bad it's going to be on Mars, but I'd like to have us be prepared.
The production of this program is made possible thanks to the support of the following and viewers like you. Well, actually, one of the elements of your talk that you gave yesterday, and also I've seen that you talk about this, is when you're in space, while you're on Earth, you still need vitamin C. Mm-hmm. And the human body doesn't synthesize vitamin C, so you'd have to be taking an awful lot of powders and pills with you, which is probably impractical. Is there some way that we can help the human body recover its ability to synthesize vitamin C using, genetic intervention?
Yeah, we can. So that's what's extraordinary is some FDA-approved CRISPR therapies involve deleting a part of the genome or reprogramming a part to turn back on a gene. The example is actually for fetal hemoglobin, which has been now an FDA-approved therapy for sickle cell disease. And it is a CRISPR therapy that goes and changes the genetic code to turn back on a gene that at one point is on when you're a fetus. It's a different kind of hemoglobin that carries oxygen in your blood, and then it goes away, but you actually just turn it back on, and that itself is the therapy and the cure. So the idea would be to look at genes in the human genome, particularly vitamin C, where even though we don't make it in our body anymore, the most of the gene that used to make it, say millions of years ago in our history, is still there. So there's these beautiful clues hiding in our DNA of what could we used to be able to do, and it could just say, well, let's just turn that back on again with a slight modification. And that would just be great for sustenance. You could then survive longer missions where you would need less supplies. You could make all of your own supplies would be the goal.
Well, would the same be true with B3?
Yes.
Because we can't synthesize that either.
Yeah,
or maybe we could before and can't now.
Well, yeah, that's another case where we could slightly modify the biochemical pathway. It's much like if you get like a set of Legos and you can build a Lego and you put it together, but if you're missing just the last piece, you have almost everything. All you need is that little bit extra. And it's like completing the pathway of biochemistry. Yeah.
So are you also able to work with the astronaut to say, we're going to make return to Earth a little bit more graceful? Because when we talk about Scott Kelly's return to Earth, it wasn't comfortable. Maybe that's being polite.
Yeah. It was very uncomfortable. So the longer people spend in space, the harder that return has been on average. Now, some people have said the inverse is also true. So if they've been to space before, when they go the second and third time, it's a little bit easier to adapt. And so, but then coming home is a bit harder. The longer they're in space so it is a bit of a trade-off. But we've seen a lot of these, what are called cytokines, these molecules that create inflammation. Whenever you get like a rash, for example, or you break out in hives, a lot of time that's driven by these kinds of signaling molecules in your blood that basically signal a stress and possible infection or damage. because the body is trying to understand, essentially this real unusual stress. And he broke. Captain Kelly broke out in rashes and had really a lot of sharp inflammation. And he
Couldn't wear clothes, apparently.
Yeah, you could basically had to walk around. Just anytime where clothes touch, he broke out in a rash. So it would have been more suitable at like a nudist colony, but that would be an unusual place to be after a mission. But it was just even clothes were uncomfortable. So it was really harsh on his skin. And so we've examined every molecule that changed when he landed. And we now have a list of, we could target them, but the big question for our lab and for the field broadly is, we see a lot of changes, but how many of them are adaptive versus maladaptive? Maybe this is what the body needs to do to acclimate to gravity, and we shouldn't stop that. Even if it's painful for a day, it may be required. But it's like most things. We'd be nice just to attenuate it. So maybe it's like if you have a fever, the body's responding to an infection. It's a natural response, but you don't want 106 degree Fahrenheit fever. You want a little bit of fever to prime the immune system, but we don't want it to be damaging to the body. So that's what we're trying to better understand is could we just bring it down a little bit?
Well, one of the other things, and I can't quite get the image of it out of my mind, is you said that his ankles swole up like the size of basketballs.
Yes, yeah.
I'm sure that he would say, I don't want to have to go through that again.
Exactly. Yeah, it doesn't. I mean, no one wants to have that degree of swelling, inflammation. It was just very painful. So we, flight surgeons at NASA are phenomenal, and they're tracking this, and there's other space agencies now also preparing for these longer missions. So we want to avoid basketball ankles for sure.
The production of this program is made possible thanks to the support of the following and viewers like you. So when you take a look at the complexity of what it is that we're trying to do, like to be able to put somebody out into space and keep them there for years, how many different elements from a genomics perspective are we talking about? I mean, we talked about vitamin C and telomeres and a bit, but what about the increased levels of carbon in the environment and on and on? And the human body so complex, it's adjusting to environments that are new to it. Like, how complicated is this task that you've taken on?
It is one of the most complicated challenges probably for medicine and for science, but it is mostly because there's so much we still have left to learn. We don't know every species of microbe that's on our skin. We know most of them, but not all of them, or even how microbes adapt to the space station, how your immune system responds to changes of the microbes in your gut or the space station or your spacecraft, what happens to your immune system, what happens if one of your crewmates gets sick and then gives you a virus. We try to avoid that, but that can happen. So, a lot of the dynamics we're just learning for the first time. And also just, to prepare for longer missions is really one of the hardest parts. If you're in low Earth orbit in the space station, if something goes really wrong, you can come home.
Quickly.
Yes, quickly, within a matter of, really less than an hour or two. But if you're deep in space or around the moon or on the moon, you're on your own. So that is one of the key hazards that we're looking at in NASA and other space agencies is how do we prepare for autonomous medical care and then have people be self-reliant far from Earth?
Well, that has been the human experience, hasn't it? For the majority of our time on Earth, we've been on our own, and the body has adapted.
Yeah.
Do we start to then say, well, if you're the child of parents and grandparents who are astronauts, you're probably more likely to be genetically in sync with what's going to be needed to survive in space? Do we have to now start making those kinds of choices? Or is it something that we go, okay, you want to go into space, we're going to help you get there so that you can survive?
Probably more the latter. I think what's interesting is, because the selection process for astronauts has been so strict and included a lot of military pilots, civilians weren't originally allowed to be astronauts. It's an unusual group of people that are just otherwise pretty healthy. So there's no known genetic basis for why they became astronauts. So it wouldn't like the kids of astronauts would have necessarily any advantage. But more importantly, even if there is an existing genetic disadvantage, let's say a high risk of a heart attack, what's called hypertrophic cardiomyopathy, or some other risk of a severe disease in flight or a vasculature problem. If you have a genetic problem today, as evidenced by some of these CRISPR therapies, you could fix it in theory. If you really wanted to go to space and had a clear, obvious need that something should be corrected, that's not possible. So the fatalism of our prior genetic states, say of 50 years ago, like you're born with what you get and that's your DNA, and your family history dictates your genetic future, is no longer true. So there is a liberty towards suddenly the genetic shuffle you got as an embryo is now one that you can modify, which is extraordinary.
So there's two other elements that I think about. You talk about like the microbes that exist like within the space station and anywhere else that you go may not be present here on Earth at all. Mm-hmm. How do we even anticipate what the body is going to need to have to be able to encounter those and survive contact?
The microbes in space evolve because all organisms evolve wherever they are, and so we've published and characterized some strains that are technically different species. They've changed enough in space that they are genetically distinct from what you'd see on Earth, but they still are functionally mostly the same so far. So we haven't seen some new capability where the microbe from the space station suddenly can process metal, or suddenly it can make gold, or do something really strange, or become a greater risk. There is possible a faster evolution that's occurring from the radiation, but nothing that appears to be a significant risk for astronauts. But they will be different. And if you go to the moon and you have people living on the moon for months and years, the organisms can and they will evolve and they will change. And so at some point they'll be pretty distinct.
So this will be an ongoing, never-ending study. The last thing is there's a higher level of carbon monoxide within a confined space like that. What happens to us, and how do we prepare for that, especially over the long term?
The gases, at least for the space station, are very well regulated. So they track carbon monoxide, carbon dioxide, they track oxygen levels and try to keep them pretty stable, and any risk to be pretty low. But they do fluctuate, and importantly, in a local sense, you can get small clouds of carbon dioxide that stay in front of your face that we can see sometimes in the blood work. So we think there is this dynamic of a little bit of carbon dioxide, which if you have too much CO2, you get a headache, you get sleepy, you get a bit tired. So it is not good for performance to be in a
can make you a little drowsy too.
Yeah, which that's not good for trying to move a spacecraft or a dock to a space station. So there are some risks that are tracked for the atmosphere, but it is some of it is just having a lot of fans is when the space station is very loud. Some of it is because there's a lot of airflow from the fans, basically.
So what's the one thing that right at the moment you're looking at going, okay, this is my next breakthrough?
I think a lot of what we're working on now is using microorganisms and organisms broadly, but to capture every atom that we can from a system. So the most obvious and I think glorious example is the space station reclaims about 97 and 98% of all of its water, and does it with extreme efficiency. So that if we could have that done for, say, nitrogen that's in urine or in urea, or phosphorus, or all the carbon dioxide is mostly vented into space. But we're looking a lot now at organisms that can actually process carbon dioxide, like cyanobacteria, and make oxygen, and think about ways to prepare little capsules that could maintain your environment and process all of your atoms. So if you're on Mars or Moon, there's autonomous systems that help you do that. So we have some good strains that we found that we're working on now, and also trying to take other lessons from other radiophiles, creatures that survive radiation, and taking those lessons to human cells with purified proteins and genes. Yeah.
It's a really exciting time, isn't it?
It's the best time in genetics and science, I'd say broadly. It's a wonderful time.
Well, thank you. Thanks for giving us an insight into the exciting world in which you live.
Yeah, well, my pleasure. Thanks for having me.